Enzyme and Antibiotic Resistance by
Bella
and Dovey. Our original experiment was with rattlesnake venom and its antivenom, where we explored how temperature affects the activity of the enzymes involved. We wanted to see if temperature could speed up or slow down the process of neutralizing the venom.
Our experiment. How does pH affect the time it takes for the enzyme to break the polymer into monomers?
Our hypothesis is that vinegar will speed up the process.
Our control group was the tap water.
Our experimental groups were vinegar and bleach.
Our control variables are the temperature of the water, the amount of Benedict's solution, the amount of enzyme, the amount of starch, the amount of each independent variable, and the amount of time the enzyme sits before going to boiling water.
The independent variable was bleach and vinegar, and the dependent variable was the time it takes for the tube to change colors.
Before we talk about the specifics of our experiment, let's first go over what enzymes are.
Macromolecules are the biological molecules that make up cellular structures and make function possible.
Enzymes are proteins. Proteins come in different shapes and sizes, and each type has a unique role in the cell. The three main categories of proteins are fibrous, globular, and membrane proteins. Enzymes fall under the category of globular proteins. Globular proteins are typically more dense and round-shaped. Generally, they have a more irregular sequencing of amino acids. They are less durable during changes in temperature or pH.
The first macromolecule is carbohydrates. The monomer is monosaccharides, the polymer is polysaccharides, and the function in a cell is energy storage and structural support. The second macromolecule is proteins. The monomer is amino acids, the polymer is polypeptides, and the function in a cell is enzyme activity, structural support, transport, and signaling. The third macromolecule is lipids. The monomer is fatty acids and glycerol. The polymer is triglycerides and phospholipids. The function in a cell is energy storage, cell membrane structure, insulation, and protection. The fourth macromolecule is nucleic acids. The monomer is nucleotides, the polymer is DNA and RNA, and the function in a cell is genetic information, protein synthesis, and energy carrier. Carbohydrates, monosaccharides, the simplest form of carbohydrates, such as glucose, fructose, and galactose. These are the building blocks of larger carbohydrate structures. Polysaccharides, long chains of monosaccharides linked by glycosidic bonds. Common examples include starch, glycogen, and cellulose. Lipids, fatty acids, and glycerol. Fatty acids are long hydrocarbon chains with a carboxyl group at one end. Glycerol is a 3-carbon alcohol that combines with fatty acids to form lipids. Triglycerides, phospholipids, and steroids. Triglycerides are formed by 3 fatty acids bonded to one glycerol molecule. Phospholipids have 2 fatty acids and 1 phosphate group, crucial for membrane structure. Nucleic acids. Nucleotides are composed of a nitrogenous base, adenine, guanine, cytosine, thymine, and uracil, a sugar, deoxyribose in DNA or ribose in RNA, and a phosphate group. DNA. DNA consists of 2 complementary strands forming a double helix. RNA is typically a single-stranded and used for protein synthesis. Proteins. Amino acids. There are 20 different amino acids that form the building blocks of proteins. Each amino acid contains a central carbon, an amino group, a carboxyl group, and a variable side chain. Polypeptides. Amino acids are linked through peptide bonds to form polypeptides. These polypeptides then fold into functional proteins with specific shapes and activities. Protein folding and enzyme structure. After translation, the newly synthesized polypeptide chain begins to fold into a specific 3-dimensional shape. The way a protein folds is determined by its amino acid sequence. The specific properties of the amino acids determine how the protein will fold. Hydrophobic amino acids tend to be tucked inside the protein, away from water, while hydrophilic amino acids are on the outside. The folding is crucial for the function. For example, enzymes have active sites formed by their unique 3D structure. The active site allows the enzyme to bind to its substrate and catalyze chemical reactions. If the protein doesn't fold correctly, the active site may not form properly and the enzyme won't function. The active site allows the enzyme to bind to its substrate and catalyze chemical reactions. If the protein doesn't fold right, the active site may not form properly. The most fundamental determinant of protein folding is its amino acid sequence.
Each amino acid has distinct chemical properties such as polarity, charge, size, and hydrophobicity. There are 20 different amino acids and each has its own chemical behaviors. Gram-positive versus gram-negative. Gram-positive have a thick peptidoglycan wall and no outer membrane, versus gram-negative have a thin peptidoglycan layer, an outer membrane with lipopolysaccharides, and more resistant to antibiotics. Here you can see the gram-positive having a thick peptidoglycan wall and gram-negative having a thin layer.
The peptidoglycan wall allows the cell to resist pressure from water. Penicillin prevents bacteria from building their peptidoglycan layer. This causes the bacteria to burst under pressure.
Let's look at how this looks on a molecular level. Peptidoglycan is made up of small building blocks, each composed of 2 sugars. The sugars are assembled into chains, which are then cross-linked via the peptidoglycan peptide bridges. These enzymes are also known as the penicillin binding proteins. These enzymes create the cross-link between chains, helping make the peptidoglycan. Antibiotics block this enzyme by making a direct bond to a key serine amino acid in its active site.
Beta-lactam ring. The active portion of penicillin is a beta-lactam ring. It is chemically reactive and opens up to form a bond. This then inactivates the enzyme and prevents proper formation of the peptidoglycan matrix.
Antibiotic resistance. Bacteria cells adapt to survive. Bacteria develops resistance mechanisms.
Antibiotic resistance at the cellular level. Beta-lactams target cell wall synthesis. Beta-lactam antibiotics inhibit enzymes involved in building bacterial cell walls. Penicillin binding protein. In MRSA, PB2A is a mutated protein that doesn't bind to beta-lactams effectively, allowing the bacterium to continue building its cell wall even in the presence of the antibiotic. Effect. Bacteria with PBP2A can survive and grow despite beta-lactam treatment, rendering the drug ineffective. Antibiotic resistance at the population level. Gene transfer mechanisms. Conjugation. Bacteria can transfer resistant genes through direct contact, sharing plasmids that carry the resistance genes. Transformation. Bacteria can pick up free-floating DNA from their environment, incorporating resistance genes into their genome. Transduction. Bacteriophages, viruses that infect bacteria, can transfer resistance genes between bacteria. Colonial expansion. Resistant bacteria multiply, leading to the spread of resistance traits in the population. Horizontal gene transfer. Resistance genes can move rapidly between different bacterial species, spreading resistance quickly.
Why antibiotic resistance is a major health concern. Increased treatment failures. Antibiotics that were once effective become useless, making infections harder to treat. Longer illnesses. Resistant infections can lead to longer hospital stays, more intensive treatments, and higher medical costs. Limited treatment options. As resistance spreads, fewer effective antibiotics remain, leading to the need for a more toxic or expensive alternatives. Now let's go back to our experiment. We use pH to slow down or speed up how enzymes work. We use bleach as a basic pH and vinegar as an acidic pH. We found that enzymes work slower or don't work in more acidic environments. You can pause here to see all the measurements that we use in our experiment, but in a nutshell, we added starch and enzyme to 3 test tubes, one with tap water, one with bleach, and one with vinegar. After 5 minutes, we added Benedict's solution and heated the tubes. At a pH of 12, the bleach, the reaction occurred in 2 minutes, and at a pH of 6, which was the tap water, our control group, it took 4 minutes, and no reaction occurred in vinegar with a pH of 2. The more basic the pH, we found that the faster the enzyme worked, showing that pH affects enzyme activity.